Hi, I'm David Sebesta. I'm KromaTiD's Chief Commercial Officer, and I appreciate the opportunity to share with you a little bit about our technology and our genomic structural analytical services. Our company is headquartered in Longmont, Colorado, which is near Boulder, and our offerings are based on our platform called Directional Genomic Hybridization, or DGH. Now, I'm not going to focus too much on how DGH works today. Happy to follow up with anybody who wants to get into the chemistry, but I'm going to rather share some specifics about the types of projects that we do for our gene editing and gene therapy sector customers. So DGH enables direct visualization of structure, and in this way it differs from sequencing or PCR-based approaches. We're gathering data from many single cells as opposed to pooled extracted DNA. We're scanning genomes on a cell-by-cell basis using engineered probes and a specific sample preparation, which leads to easily interpreted images. Any out-of-place signal translates to the direct observation of a structural change versus the reference genome. So up here we have the flavors of DGH, so to speak, each optimized for different applications. We have the targeted DGH Insight for small targets, transgenes, viral integrations, inserts, or edit sites. We have a DGH Screen for an unbiased assessment of larger rearrangements throughout the genome. And these unbiased screen-based assays, they can focus on a single chromosome, a subset of chromosomes, or the whole genome. And much of our custom assay work really involves combinations of targeted and unbiased probes, actually. So in the interest of time today, I am not going to talk about our fanciest development stage approach, but together these assays form a very versatile structural genomic tool set, and when combined with sequencing, promise to provide the basis for the most comprehensive structural analysis available to gene therapy developers. We launched our gene editing based services in about 2018. However, as an unbiased technique capable of detecting inversions, translocations, and other rearrangements, DGH is ideally suited for any application where genomic structural changes are of interest. And in fact, this whole thing started in the radiation biology field, where there was a need for a reliable technique for detecting and quantifying damage attributable to ionizing particles from deep space travel or other sources of radiation. And a lot of the early funding for the company came from NASA. To this day, we still team up with our scientific co-founders and NASA. We analyze every astronaut that visits the space station for chromosomal damage. And our co-founder, Professor Susan Bailey, was one of the PIs on the NASA twin study, which received a lot of attention a couple of years back. However, mostly these days we're focusing on measuring the intended and unintended rearrangements associated with cellular engineering processes. And a recent paper clearly articulated the unmet need that we're beginning to fill with our technology. So there was a study commissioned by the WHO that was led by Dr. Zhao at the NIBSC, and there were many other institutional and industry co-authors as well. And it really comprehensively studied the outcome of viral-mediated integration using the gold standard PCR and sequencing-based methods. The conclusion of that study was that, due to limitations in sensitivity, quantification, data interpretation challenges, that there is a real need for orthogonal methods. And there are a number of ways in which KromaTiD's DGH is now fitting into gene therapy developers' workflows, and I'm going to go over some of those now. So this way too busy table, I'm not going to dwell on every point here, but we offer services to the gene therapy field based on targeted DGH Insight assay designs, based on our unbiased DGH Screen assay designs, and like I said before, combinations of these two in a single assay. With the targeted Insight, we are making single cell measurements of location and orientation of the target or targets in an assay, and that can include genome-wide distribution of integrations, for example. We also make a lot of targeted probes for genes or edit sites within genes and such, and we use probe colors as well as other tricks to extract structural outcomes for targets. Depending on the sequence of the target, we can design probes with lower limit of detection in the 3 to 5 kilobase pair range, and we think we'll be down to kilobase levels soon enough. Of course, there's no upper limit on the size of a given targeted probe. In contrast, unbiased DGH screen paints provide the ability to detect rearrangements in single cells de novo. You don't need to know where to look to observe rearrangements, such as translocations and inversions, as well as gross structural and morphological events like aneuploidy, micronuclei, and chromothripsis. The application for these measurements that are in use by our customers span from fundamental research questions about on and off target outcomes of editing systems to quantitative confirmation of the extent of unintended events, to screening assays, and even QC applications within process optimization and manufacturing of engineered cellular products. DGH assays lend themselves very well to kind of comparative testing, before-after passages, one editing system versus another, pre-existing variation in a subject versus the reference genome, and so on. As for sample types, we can design targeted probes against any published genome and have done not only humans, but also non-human primates, murine, canine, feline, porcine, probably others, genomic targets, any published reference genome. As a synthetic method, the design, manufacture, and qualification of our probes is actually very cost-effective and efficient. Finally, we have done many dozens of projects for dozens of customers and have worked with T cells, iPSCs, HSCs, many cancer cell lines, blood cells, tissues, CHO cells, and I'm sure there's others. So I'll briefly show just a little slice of data from a study that's leveraging both DGH Insight and DGH Screen in the assay design. And this kind of illustrates some representative ways we approach study design and how we parse up the data. So this system is a targeted integration of a 10 kilobase pair transgene. We have green probes bracketing the target integration site, a yellow transgene probe, and we're monitoring random events, on unedited chromosomes using our dosimetry paints on chromosomes 1, 2, and 3. The scoring approach for this study will yield quantitative information on the prevalence of on and off target integration of the transgene and the distribution of integration events by cell. So we're showing the image for one of 200 cells that we counted in the study, and specifically, this is a cell that showed on-target integration of both homologs. You can see the co-located yellow and green signals here in the blown-up box. In the edited sample population overall, we saw a lot of off-target integration. We saw on-target plus off-target. We saw on-target in only one of the two homologs, and we saw a lot of no integration at all in about half of the cells. So we worked these samples up in several other ways, including analysis of the relative intensity of the integration signal to estimate copy number. We're happy to drill deeper into the study and the potential of the approach with any interested parties, of course. There is another embodiment of our technology that I wanted to highlight today as well, and we call that Pinpoint Fish. So, the DGH process requires dividing cells, and of course, not all samples are dividing. We can also employ the targeted probes in all kinds of samples traditionally analyzed by fish probes, including tissues and dissociated interphase cells. Because our probes are engineered to bind only to unique sequence, background interference is basically non-existent, and the signal to noise is great. And that's kind of how we came up with the name of Pinpoint Fish. But it's just wanted to point out that we have options for the analysis of structure within non-dividing cells. It's just a little bit less orientation and structural data than comes from DGH. And as a final slide, I will just kind of walk through how we get started on a project. So everything begins when a customer provides us with target information and then guidance on what they care about measuring. So if we have a custom probe that we require, we can perform a design review and sort of confirm the suitability. We'll usually have, at least 1 or 2 technical scope discussions where we flesh out exactly what the measurement objectives are, how many controls, active samples to include, what level of sensitivity is required, so we know how many cells to count. And then from this stage, we generally have enough information to generate a quote. Once signed, we provide a probe design description for approval by the customer, and then that triggers the manufacturing of the probe. It's really critical while the project is underway that there is a lot of ongoing communication, and we structure project calls with the customer to really just put the entire thing in the best position for us to get the project right the first time. What we do mid-project, we look at a representative fraction of the data from the samples. We'll generate a proposed approach to the scoring protocol. We'll get together with the customer for an interim data review meeting. An official scoring SOP will kind of fall out of that. We provide that to the customer for approval, and then we'll finish up with the rest of the scoring. And then, of course, the deliverable, a report breaks out the findings, depending on the scoring approach. There's a narrative description of the outcome, there's tables, there's graphs, etc. So we're very proud of the fact that virtually every customer we've worked with over the years has come back for additional projects. We get a lot of positive feedback on our collaborative approach. We're very passionate about what we do. We would love to talk with you about how we could help you solve your problems, and hopefully be hearing from some of you afterwards. Thanks again.